Hi, everyone. Welcome to OsteoBites. Thanks so much for joining us today. My name is Christina Itoma, and I'm mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Alanna Church from Boston Children's Hospital. She's one of the co-chairs of the AACR Special Conference in Cancer Research, Discovery and Innovation in Pediatric Cancer from Biology to Breakthrough Therapies, that just wrapped last week in Boston, and we are delighted to have her join us to share highlights and key takeaways from the conference. Thanks so much, Dr. Church, for joining us on OsteoBites today. Everyone, please feel free to submit questions during the presentation. This will really just be kind of a more casual chat. So any questions you have, just put them in the Q&A in Zoom, and we will share those with Dr. Church just as she goes. So a little bit more about our guest today. Dr. Church is a molecular and pediatric pathologist at Boston Children's Hospital, where she is a founder and associate medical director of the Laboratory for Molecular Pediatric Pathology. And she's an assistant professor of pathology at Harvard Medical School and the program director for the Harvard Molecular Genetic Pathology Fellowship and the incoming chair of clinical practice for the Association for Molecular Pathology. Her clinical and research work focuses on bringing molecular testing to the clinical care of children with cancer. And through institutional projects like the Profile Study and the GAIN Consortium Study, she has profiled thousands of children's tumors and has used these results to make real-time impacts on their diagnoses and treatments. She's involved in national initiatives to improve the quality and access to molecular testing for children with cancer, including the NCI-funded Count Me In study through Dana-Farber and the Broad Institute, the National Comprehensive Cancer Network, the National Institutes of Health, and the Children's Oncology Group. And we are so fortunate to benefit from her expertise on our MIB Agent Scientific Advisory Board. We're thrilled to have you on OsteoBites, Dr. Church. Just an announcement before we get started, actually a couple, as we roll into fall, we have a lot going on. We just opened our Outsmarting Osteosarcoma 2026 grant cycle. Letters of intent are due October 24th. And then if accepted, invitations for full applications will be sent out in early December. And full applications are due January 25th next year. So I'll put a link in the chat where you can find the program guidelines and the link to apply. Also, our annual Factor Conference will be in Columbus, Ohio, June 25th to 27th next year. And we're opening abstract submissions next week on October 6th. Submissions are due January 9th, so a lot of lead time to think about it. Abstracts will be considered for both oral and poster presentation. And so just wanted to plant that seed in case you're interested in sharing your work at the conference next year. So let's go ahead and we'll get started today. Dr. Church, if you want to go ahead and start sharing your slides.
Thank you so much, Christina, and thanks everybody for being here with us today. I'm excited to get to talk to you about this conference that we just had last week. Are you able to see my slides, Christina?
Yes, that looks great.
Okay. So the conference was local for me. It's in Boston, and it was nice on a lot of levels. But one is that, of course, we have a big community of people involved in pediatric cancers in Boston. So we had a lot of local representation, including from many of our trainees and junior faculty here. And we also had a lot of people actually from all over the world. And I'll talk a little bit about the global aspect of it. So this was the focus of the conference this year. This is the first time the AACR has just switched to having this as an annual meeting. So we're gonna hopefully soon release the location for the meeting next year if anyone's interested. And I think there's a lot of interest in this space, and that's the reason they're sort of investing in us and in this way. And so I'm going to try to focus some of my talk on what's most relevant to the osteosarcoma community, although of course this was focusing on all pediatric cancers. And since I was one of the organizers, I can say this with confidence that we really tried to represent hematologic malignancies, brain tumors, and solid tumors. So there's like a smattering all the way across. I would love to hear questions. I actually was just saying this to Christina before we started. I don't love these ones where I just talk and like there's no back and forth. So please put your questions in the chat. I should also mention that since I'm a pathologist, I do a lot of work on molecular and genetics. So can answer all of your questions there. I'm not a drug discovery person, so I will do my best to represent that side of it. And there was a lot of drug discovery talk in this meeting. So we were three co-chairs of the conference. That's me, and then on the left is Charlie Roberts from St. Jude. And in the middle is Meredith Irwin, who's from the Hospital for Sick Children in Toronto, in Canada. And I actually used some of Charlie's notes from his conclusion speech while I was putting these slides together. So huge thank you to both of them. And they were so great to work with. And the AACR team is really amazing. So I know I'm kind of preaching to the choir here, but like this is such an important area. Pediatric cancer research and clinical care. There's a lot of movement in the space right now, which I think is really exciting. The conference was about 250 people. People really like hung in for the whole time. You know, they were there at the beginning all the way through to the last day, which was on a Sunday morning. And there's really good energy in the crowd. I think we feel that we're at a turning point now of really getting into more targeted therapies and that the research is moving quickly. And there was a lot of great discussion about collaborations. You know, I think in pediatric cancers, we're sort of good collaborators because we all have to be because every cancer is rare. And this has been the practice for so long. But really kind of leaning into that. And also, you know, there was some good discussion about partnering with you know, the drug development community, pharmaceutical industry, but also really about families and patient advocates. And I spoke about that in my session, and I talked about you guys at MIP, but there was other people that sort of referenced that as well. So there was a good global perspective. So we began on the first day of the meeting by having our two award winners. Sean Lee came from Singapore and spoke to our group. He focuses really on hematologic malignancies. And he gave a beautiful presentation as our first keynote of the whole meeting and talked about the work that they're doing in Singapore to do really comprehensive molecular profiling, not just for the patients in Singapore, because they have a more kind of national healthcare system. So they're able to do things very uniformly there, but they're also doing a lot of outreach to the neighboring countries, even where they're not able to do, you know, run clinical trials necessarily inside of those countries, but they can have the samples sent to them at their lab, do the molecular profiling, and then share the results. And I think this was a theme of a lot of what we heard in the conference is that really now, and this is kind of getting to my area, the molecular profiling that's underpinning all of the treatment decisions. You know, I think the people in heme malignancies have been well ahead of us in solid tumors in this capacity. So they've, you know, been doing a lot of molecular profiling for many years and have a very, you know, granular kind of risk stratification process that they put their patients through to decide which therapies they should go on. And some are targeted therapies and some are more sort of traditional chemotherapies, but they're all, you know, very well characterized. And so he's having a big impact, you know, not just on the patients in his country, which is already, you know, a big feat, but also really at a global level. Dr. Nana Jaboto from McGill and Montreal had a more kind of mechanistic sort of developmental talk where she shared the work that she's been doing in pediatric brain tumors. And she really discovered this kind of histone modification that we now know to be a driver in a lot of pediatric cancers that wasn't known several years ago. And so that has led to kind of like a whole new class of brain tumors for our pediatric patients. And, you know, it's not exactly targetable yet, but it's something, you know, now that we know the driver, that's something that people are working on targeting and is a target for drug development. Christina, you'll interrupt me with the questions, right?
I will.
Don't be shy. Okay. So a lot of nice talks about development. So as you can imagine, when we're thinking about developing new therapies, sort of like the work I just described about histone modification, there's a lot of really basic biology work that needs to be done to understand what the drivers are. So we had discussion of all of that sort of from end to end. So a lot of developmental biology, discussions about chromatin regulation and epigenetics. So if we think about DNA as being like the blueprint of like how to make a person or a banana or whatever, you know, epigenetics is sort of the layer on top of that controls which genes get turned on at which time. And it's a very important, of course, as you can imagine, for what actually happens inside of the cells. And it's an area that I think we know less about than we do about DNA, even though there's still plenty we have to learn about DNA. So a lot of people working on that, and there are new techniques to do methylation profiling, which we heard about in the liquid biopsy session too. And so this is another area that people are really hopeful when they're thinking about drug development. So there are new therapies emerging. This is where don't ask me too many hard questions about these because I don't understand them as well as some. Certainly the people in the conference. But there are these new classes of therapies that are getting approved, and none of them really specific to osteosarcoma yet. But I think it feels to me, as someone who has been in this space for a little while, that there's an acceleration of new options that seems to be coming through a lot faster than it has over the past few years. So a lot of excitement about these targeted inhibitors in brain tumors and in leukemia. A lot of excitement about CAR T immunotherapy. And again, this is more focused on leukemias and neuroblastomas at the moment, but it's a great new approach that's really different. And that was one of the themes that sort of kept coming out throughout the meeting, is that we're not just looking at doing minor variations on the chemotherapies that we've had for so many years, but really trying to do something new. Christine, I saw you unmuted. Did you have a question?
Oh, yeah. I was just going to say, so, I feel like when more patients were doing molecular testing, and there was kind of this idea of like, oh, it's not, it's kind of like tumor agnostic. It's kind of more about what the, mutational drivers are. And so for some of these emerging therapies and agents, is there any thought about kind of applying them kind of more based on the mechanism of action, and if there are commonalities across disease types versus looking at it for a specific disease? So, for example, was it larotrectinib? Was that the NTRK
Yes.
inhibitor?
Yeah.
Good
Larotrectinib is
example of
like
that.
the perfect example. Yeah. Yeah, I mean, I think that, and there was some discussion around this too. There was a panel where we were talking about, new strategies to try to like think about things in a different way. And I think having tumor agnostic approaches is what everybody would like to do and has been very successful in pediatric cancers, because again, we have so many rare tumor types and everything's so unusual, right? So you don't want to like focus too narrowly, to use one drug in only a specific area. So of the list that we can see in here, EZH2 inhibitors, potentially could be used across different tumor types. Larotrectinib, which is the example that you gave, is a great one because it's a drug that works really well. They sort of, even in the drug development time and while they were going through clinical trials, pulled in a lot of different tumor types. And so they were really the first drug approved in a tumor agnostic fashion, right? So it wasn't like a drug for this disease. It was a drug for any cancer with this alteration. And so that alteration for larotrectinib is NTRK fusions. So if you had an NTRK fusion, and if an osteosarcoma patient had an NTRK fusion, they would potentially be eligible. Some of the other ones, they're sort of classified by, I mentioned CAR T immunotherapy and GD2. So the reason that really kind of works well in neuroblastoma or the CD19 in leukemia is because leukemic cells present those proteins. So that's what the immunotherapy is going to. So for that one, it kind of doesn't like exactly work to make it broad. If there was another tumor type that kind of presented the same protein that could kind of act as the antigen, that would work. But the approach is one that can be kind of modified, right? The immunotherapy or the CAR T approach could be applied to different tumor types, right? So if we were thinking about osteosarcoma, what is something that is expressed on the cell surface of osteosarcoma cells that could become a good target for immunotherapy? And those are the kind of conversations that people were having about these drug discovery elements.
Great, thank you.
We heard from Jack Shern, who was with us from the National Cancer Institute, about the COG Molecular Characterization Initiative. He's a great speaker, and his focus of his talk was not just about the MCI in general, but he really zoomed in on sarcomas, so more relevant to our crowd. He really emphasized again, as we've talked about many times, that sarcoma represents a really diverse group of tumors, and there's not like one solution that can be applied. And I think it's something that, I think we know, but it's great to hear it again and to hear the data behind it, and to have somebody show in such an elegant way the data behind it. He discussed, as some other people did as well, that liquid biopsy or circulating tumor DNA is something they're pulling into the MCI, and that's something everybody's excited about, right? Because we want a new, better approach to do molecular profiling, and to really, I think the space where it's been proven best so far is in monitoring, right? So like showing that the level of ctDNA in the patient's blood is a reflection of how well they're responding to the treatment that they're getting or not, and the idea there is that if you can tell that a patient is doing well on the therapy they're on, great, and maybe they need like less total therapy, and that patients who aren't doing well on the therapy again, may need more, they may need a different treatment. And so that's the way that we're sort of thinking about liquid biopsy in a pediatric cancer setting for now. One of the things that Jack spoke about that I thought was really interesting, and that sort of connects to some of the work that I'm doing that I'll share about a bit, is that because this is through COG and the NCI, there are opportunities for children and families from a lot of different hospitals to enroll. So he had this chart of like enrollment, and many of the people that have enrolled have come from really small institutions, places that are far away from big cancer centers like ours, and they're enrolling they've only enrolled one child ever, in this study, but there are many of those. And I think it feels a little surprising sometimes. We, in the bigger centers kind of get this idea that everybody ends up, with us or at Texas Children's or, one of the other big hospitals, but of course that's not the case. And there are many patients and families who are in lots of different communities. So it's really special to be able to have this initiative that is touching so many families across the country. And this is US and Canada also, I should say, just not just the US. So we're in the kind of middle of this MCI time right now. The data that they're showing is really great, right? They're able to identify drivers in a lot of cases, and they're generating a huge and amazing data set that, is intended to be for all of us in the research community to use. And because they're doing it through COG, they also have, they have the pathology slides, like the H&E slides that they photographed, and those are viewable. And that this data will just get better and better as we kind of move through the study. So that's a huge and amazing output of this whole initiative. Did you have a question?
I love these guys. I feel like it's such an amazing resource, CCDI.
Yeah.
So one thing though that I am very anxious to like get an update on is the Molecular Characterization Initiative, the MCI. I know they recently started with Ewing sarcoma, right? I think they added,
Yes.
and
Yes.
and I'm kind of anxiously waiting for them to add osteosarcoma to
Yes.
the list of diseases.
Yes.
Was
I think that would be
there
great.
Yeah,
Yeah, that would be
they
great. And I
talked
have
all
been,
about.
yeah, I have been involved in some conversations about this because I think, sometimes people, as you know, are reluctant to include bone tumors because of the decalcification, and they're not gonna work. So we've had a lot of experience with those samples, and we, are trying to advocate. Yeah. And of course, Katie Janeway is like super involved, and is the best advocate of all for this kind of thing. Have they said that they're going to include osteo? going forward?
No,
No,
no, no,
not yet.
but they
Yeah.
did officially announce that they are now including Ewing sarcoma, which I kind of
Yes,
feel
yes.
like, I mean, there's another bone cancer, so I feel like they've, paved the way for osteosarcoma. So
I hear
hoping,
you. Yeah. Yeah, and yeah, they're doing such an amazing job.
they are. It's a great group.
Yeah. So next, this is same Ajadi's photo. I have to share this analogy that he he provided to us. So I have never heard anyone say this before, but I love it. I've already told several people. He was beginning his talk by, mentioning how diverse pediatric cancers are, and he said the way he explains it to people when he's kind of out there in the world is that adult cancers are different from one another, but not that different. They're like a bunch of cows in a field, and that the pediatric cancers are like the London Zoo. He's from London, and it's so true, right? It's really a great way to like easily convey the complexity that we are facing when we think about the pediatric cancers sort of as a whole. Yeah, so I have to give him credit for that analogy, which I'm totally using and will
Yeah,
continue to use.
it's great.
Yeah. And he spoke about the experience that they've had in the UK, and they have, of course, like a different system. So they, through the National Health Service, have the opportunity to do their molecular profiling in, at a national level. And they have been using whole genome, whole transcriptome. Sorry, my dog is barking. Whole genome and whole transcriptome approaches there, and sort of comparing how different some of the different results that they're identifying, with that kind of more holistic approach compared to a targeted approach, which is what many of us in the US are. Sorry, just
I mean, we've advanced, so technology improves so much so quickly. I mean, it used to be that like WGS or WKS would take longer, be more expensive, but are we kind of at a point where that is feasible for each patient versus having to do a targeted panel?
I don't know. I think about this a lot. I think and we were just discussing it, of course, as you can imagine, at this meeting. When we think about whole transcriptome, the alternative to that is an RNA panel where we're looking for fusions. And we are using in our lab, so I should say this as a caveat, we're using a targeted panel, and we found that it's been just really sensitive. Like for the fusions that we know about, it's very quick, it's reliable, it gets them. The whole transcriptome approach, it allows more like discovery of things you've never seen before, but it's not as quick to use. It's not as reliable for the alterations that we do know about. And those are the ones usually that you want to kind of return clinically in a quick way, right? You don't want to have an NTRK fusion kind of sitting around waiting for like a big complicated analysis. So there is interest in bringing in whole transcriptome over time, but I'm not like sold myself yet. The whole genome approach, similarly, the targeted panels, have been constructed in a way that they work really well and they're very reliable, where the whole approach may miss things. So again, I'm not like all the way sold, but Sam did a great job kind of bringing me along the road because he showed a few cases where they, of course, they identified things that were unexpected, and and could be transformative. And they published a nice paper last year in Nature Medicine about this. But so there's a lot more detail than what I can recall and sort of give to you today. But when somebody asked him in the audience, like, who is this for? Kind of to your question about like, if we've been doing panels, how do you know which group which group of patients should be getting this kind of more holistic approach? And his answer was, you don't know who it's going to be for. And all of the patients that were on his list where they had found something were ones where you wouldn't have guessed that it would be useful. So it's kind of for everybody in the sense that like, you don't know who is going to benefit from it. Yeah, I think part of my kind of hesitation in saying we're ready for all that is also knowing like he has a great group and a big team that can really work with that data and analyze it. It's hard to do that in a clinical lab where we're more constrained. with like, our time and resources.
Yeah, I was going to say, actually, if a patient wanted to get that done today, would they have to go to a commercial lab? Um, you know,
St. Jude does whole transcriptome,
okay.
and they do a very whole approach. So I think you have to be on a clinical trial to get access. I'm not sure, actually. There are a few places, and there are some commercial labs. I mean, I would also say when you're thinking about a commercial lab option, what really matters the most is who's looking at the data, right? Because there's so much data and there's so many ways you could look at it. You really want someone who knows pediatric cancers because of this new, issue. So the St. Jude group would be one that, of course, that's what they do and they know what they're doing. If you're thinking about a lab that mostly kind of services adults with cancer, you can get the kind of lab bits of it done, right? Like that it's gone through the sequencer and had those, but you really would want someone to look at the data who knows and understands pediatric cancers.
Sure. A zookeeper.
Exactly. We're not a keeper, like a friend. Yeah. Yeah. All right. So we had a very nice presentation from a parent who shared her story, and her son is still going through his treatment for leukemia. It was really amazing. There were a lot of tears in the audience as she was sharing her story. And at the end, it was very touching. And at the end, she, took some time to kind of say thank you to everybody who's in the audience, but also, to kind of remind us, and which she had done throughout the story, that the treatment course that her son has been on and is continuing on is really difficult, right? And they really want better, they want better, even, for the patients who end up surviving their treatment and doing well. It's such a rough course. And she spoke really beautifully to that, and about how he had like missed school and he'd been away from his brother. And this again brings up kind of one of the themes of the conference for me, which is that we can hopefully do a better job of partnering with our patient community over time. And I think she was there also to remind all of us why this work is so important. And I think in some sense, like a lot of people in the audience, we're already there, right? But I think it was really nice, probably especially for, we were just talking about transcriptomes, and there are people there who are researchers, even people who do pediatric cancer research every day, who are pretty far removed from patients. And I think it was a very beautiful reminder that the work that they're doing is so important.
That's great that you were able to do that. As you know, we're big, MIB agents are big believers in that, on kind of bringing the.
Yeah, you guys are the best at this, like bringing everybody together. It's amazing. So this was my talk, and I actually spoke a bit about this initiative at Factor last year. But I've been able to put together this really amazing group that I called Sprout, because you have to have like a cute name when you make a new when you make something new. And so we have this group of experts from around the US and Canada who have been working together to create guidance for our oncologists and pathologists about what molecular testing should be done for a child with cancer. And we have a full set of guidelines now that we're actually just finishing up our paper this week. So hopefully we'll be able to get it out there soon, and we'll share it with you. But we're hoping, we did a very thorough, literature review and put together all of the data. So the data that we had coming out of the GAIN study, there are, Sam Bajati's work from the UK, the Australia Zero Cancer group has done a really beautiful job. So there's a lot of data out there now that is sort of building and together shows the importance of, and the clinical utility really, of molecular profiling for pediatric cancers. So how can we translate that into guidance? Because we know from, our clinical experience and from surveys with our clinician community that people aren't not everybody's getting this done, right? Not everybody knows what to do. Not everyone is able to get it paid for by insurance. And so there are a lot of gaps. And we're hoping that this guideline will help just, and it's pretty simple, actually. We're kind of saying everybody should be getting DNA NGS. And we discuss in there sort of the approaches, right, of like targeted versus whole. And we don't come down on one or the other, but that everybody should be getting DNA NGS. Many children should be getting RNA NGS looking for fusions, and that children with brain tumors should be getting methylation-based profiling, because that supports the diagnosis for them. And so we're hopeful that, you know, having these guidance documents out there will help to guide people in knowing what to do, and also, you know, serve as kind of a push to the payers that this should be getting reimbursed. And so over time, that this will become the standard of care for everybody. And you don't have to we don't have to continue to rely on the MCI or continue to rely on projects like at the larger cancer centers to fund this work. And we emphasize that the patient partnership, and I also got to talk about Count Me In, and I showed this landing page for the Osteosarcoma project, which was fun to do. I didn't play the video because I didn't have enough time. I love to put that up.
Yeah, I love that you're doing this, but I also just love that it's very practical. You know, it's because it's not just the guidelines, but it is driving towards the coverage, which you're right is like a big barrier. A lot of people doing that, so it's so important. And I love the name and I love the logo.
Thank you. Yeah, you know, one of the surveys that we did for pathologists was asking them, like, what are they ordering? And a lot of them still are ordering kind of like our older techniques, like FISH instead of NGS when they're thinking about a fusion. And they reported that they're not sure about how it's going to get paid, right? So that the issue of payment comes up in a lot of ways. And sometimes it shows up in the sense that, like, the clinician or the pathologist who has that case in their hand, they don't want to order something that they don't know, you know, if the family's going to get stuck with a big bill that they may or may not be able to afford, you know. So it has this kind of trickle down effect that sort of leads to less testing being done, even if, you know, potentially that one would have gotten paid for. We don't know, because it's hard to predict, right, when it's going to be approved or not.
Yeah, you know, kind of something I think about in relation to osteosarcoma specifically is, you know, so we're getting closer, I think, for sure, for a risk stratification, because, you know, Amanda Mironoff is working on the CBOS consortium to really also kind of bring that to clinical practice. So there's, you know, risk stratification. You know, historically, like there's in osteosarcoma, sometimes a lot of the mutations are not druggable. So even though you have that information, you can't really act on it.
Right.
But I think, you know, the other kind of big benefit here of everyone doing it is just the data gathering. Because I also just think about where we are now, and as we advance towards this inevitable world of AI, which can be so powerful, but if we don't have any data for it to like learn from or work off of, like, really help us.
Yes.
But I'm wondering, like, I mean, so, you know, let's say we get to a point where it is kind of standard of care. I mean, one thing that's really great about MCI, or for example, is that it is kind of centrally available somewhere, all of that information.
Yes.
And I'm like wondering if there's also an effort, which, like, let's say we again get to that point where everyone is getting it done, if all that data can be collected and aggregated.
Yeah, and I think there are a lot of people working on this. It's a difficult problem, you know. In the Count Me In study, that's going to be a great source of data, right, for osteosarcoma specifically, because we actually have a very big cohort, thanks to all of you guys, of patients, and the data that's being generated is very rich data, because we have a lot of clinical data associated with the genetic data, and that's really the best kind, right? You want to, if you're thinking about risk stratification or response to treatment, you really need to have associated clinical data to know, you know, even when you're at the level of a spreadsheet, like which of these patients responded well or not, to correlate with the genetic signature. That's what like drives a good analysis, and, like, you know, can take us to the next level for a drug discovery. So I think like the CCDI initiative is an important one. And they're thinking broadly, beyond just the MCI, what they're doing a great job of sharing what they have, but also I think thinking ahead about how to create better ways for us all to do data sharing. It takes a lot of work, any lab, like we were just talking about, our labs are all so busy. We're busy doing the next run and the next run, and like to set aside time and resources to like tidy up your data and put it into the right format and send it to the right place, and then who's going to take care of it and catalog it. And there's so many issues around like data privacy, so people need to be thoughtful about that too. And it's typically like not an area that gets funded well, right? Because it's sort of like databasing, it's a little dry. But I think it's really important, especially, as I'll keep talking about that London Zoo analogy. We need it, for the zoo. Otherwise we're just kind of left like trying to solve things one by one.
Yeah.
I said I wasn't going to take up the hour, but I totally am now. So keep going.
There was a lot.
It's so exciting.
There's a lot
Yeah, I know. There's a lot going on. Yeah, so there was a whole session about ctDNA, about liquid biopsy, and very exciting. And again, sort of touching on mostly not hematologic malignancies, actually, in that setting. The brain tumor groups are doing a lot of work with ctDNA. They're using fragments of DNA from patient's blood, but also from the cerebrospinal fluid. So when patients are getting lumbar punctures, they're able to pick out bits of DNA there that can help with the diagnosis and risk stratification. And again, I think I'd mentioned this already, but there is a potential application which Brian Crompton's group has shown for osteosarcoma of tracking levels of ctDNA as predicting response. So it's more of like a real-time monitoring. Many families have expressed this kind of, and the patient, the mother who did the presentation, during this time when you start, you're giving your child this like kind of terrible treatment, and you're like, gosh, I hope this is working, and so hopefully we can kind of provide some data at that time point and know what isn't working. And potentially for predicting relapse. Sometimes when I'm talking about ctDNA and liquid biopsy, people ask, is this a way for early detection? And I usually want to say not to be like a naysayer. That would be great, but it's not. So I don't think we're there yet. But I think the real-time monitoring is a really important piece, and we're leaning into that.
How far away are we from that really becoming clinical practice?
I think we're probably within a year of doing clinical trial work. And some clinical trials have already kind of started I think within a year or two, we'll definitely have in trials where the results that we're getting from liquid biopsy will change the treatment that the patient gets. And the results of those trials will inform how standard of care it becomes, if that makes sense. The issue that we have faced when we're thinking about liquid biopsy is similar to some of the other themes that we've already talked about. There are companies that are doing liquid biopsy genomics. There are a lot of companies, but many of them are not pediatric focused. So they are not getting the right genomic targets that we would want and need. So Brian Crompton's work has been really special because he's, of course, taken a really focused look at pediatric cancers and has created a panel that works for pediatric cancers. So, we need to look at fusions, we need to look at copy number. It's so different than what you would look at for like an adult with a colon cancer or breast carcinoma. And my lab is working with Brian to bring that into the clinical lab so that we can return the results to patients, whether it's in the clinical trial or, or sort of like in a more kind of routine basis, and so that they can like use it, right? So you're not just looking backwards at a trial that's already closed and saying, did it work or not? But like, we want to show that we can use it, now today to change the patient's treatment, and that ended up with a better result. So that's where we are kind of in the pediatric cancer space of getting just at the cusp of bringing it into clinical trials. And hopefully, I mean, well, it'll become standard of care if it works, right? If it's good, which is what we want and hope. But yeah, I would say we're probably a few years away. Even if you think about what I just talked about, like doing DNA NGS is still not standard of care. It takes time for things to become kind of standardized.
Yeah, it makes so much sense though, because the machinery is there, right? Like patients are going in and getting labs regularly. Though I did just actually, as you were talking about that, think, oh wow, that might change the whole paradigm of like skin society. Like at some point, if you are also
Yeah,
in your lab results, also kind of monitoring that.
yeah,
Um,
yeah. Well, you have blood draw anxiety.
yeah, yeah,
Yes. Yeah. I mean, we're not going to be able to, take away the anxiety of the whole process in general, but hopefully give them better sooner data.
yes.
There was a very nice panel really talking about some of the challenges, like at a kind of operational level of drug development for this group. The zoo analogy keep coming back of all of these rare cancers. So, good discussion and a lot of like energy in the crowd. I think it wasn't like contentious. I think we all agree that this is an area we need to lean in on, and what can we do to like convince some of the pharmaceutical companies to take on pediatric cancer patients. We understand their reservations both from a kind of economic perspective, but also they sort of, I think, often see kids and teens as like risky, and they don't want to have a bad outcome. um so some of the things that we talked about were that also drugs that sort of get approved for adults, but they're not really as accessible for kids. And a lot of funding gaps for early trials because, there's the discussion of like just kind of hopping on to drugs that are coming through or are already available for adults. But, because the genome is so different and we're, all these unusual tumors, we need our own early trials, right, that are really focused on the unique genes of a pediatric cancer, which maybe, doesn't appear at all in adults. And that there are funding gaps. I mean, we all know we're in this really difficult moment right now with respect to research funding, but that early trials was kind of called out as being one that really is a need in this space. I'm very
Was there some
grateful to have organizations like MIB who are such good funders.
Yeah, well, I was like, because these are obviously really difficult and complex problems. It's not, but I'm just curious if there was discussion about what are some of kind of the most impactful levers that could change the space. So, for example, I feel like a lot of the legislative work has been really impactful, instrumental. It kind of solves it, gives like the carrot, does
Mm-hmm.
because of the
Mm-hmm.
right, like because it's not make a lot of money on
Yeah.
these
Yes.
pediatric drugs, but they created this model where it was like there was a financial incentive um, to make your drugs available for pediatric patients. So I don't know, was there any discussion? I'm especially curious from the pediatric partners. I'm sorry, from the pharmaceutical partners on kind of
Yeah, there
what.
weren't a lot of pharma reps there. I should say it was more kind of like the researchers and clinicians like me, kind of just talking about them. But yeah, there are some like sort of bigger initiatives, like Accelerate is one that people talked about, sort of getting together and kind of being more of a force together. I think that's probably the most important lever. Steve Du Bois gave a nice talk where he kind of showed with historical data about trial enrollment. I should also mention like that congressional act to include children really was brought about because of the patient community, right? And the voices of parents. So that is very special. But that, a lot of the trials still don't include children. I mean, he, had this graph and then there's this arrow where the legislation came in and it hasn't changed that much. So I think we need to keep I don't understand this area super well. So, I mean, you would want to ask Steve for the details, but like, how are they getting around it? I don't know. And there was also discussion, in that kind of Q&A portion of Steve's talk about how when if you kind of are pushing for a drug company to include children in their trial, the easiest is for them to include older children, right, like teenagers, and that there's a worry for people that kind of, if you think about neuroblastoma, which mostly affects babies and infants, that they're when will they be included, and they really probably need something more for them. So like, early trials that are more focused on them as a group. But I guess that's one of the concerns about like how the legislation and how the rules are applied and implemented. And it's great that people like Steve are looking at that, because I think it's important, of course. All right, I'm almost done. So we were done. There was another panel talking about sort of rewriting the rules, which I thought was really great. We've been sort of in this era the last few decades of, kind of like traditional cytotoxic chemotherapies, and then, sprinkling in more like molecularly targeted therapies over the last few years. But that all of the discussion in that whole conference was really about like how to do everything differently. Which sounded great. The ones that the drugs that people are excited about that are working well now are, we showed some examples of some of the new things. But there was good energy in the audience, and I think a lot of optimism. Yeah, I was going to say not optimism. I mean, but no, it did feel like that. Like people are feeling the excitement of kind of this space, and that there's a lot of movement, throughout the drug development kind of pipeline, from like, lab developmental biology to things actually getting to patients. There was a lot of talk again in that sort of rewriting the rules about combined approaches. So, when we were thinking about even like larotrectinib, which is a great example because it works so well. But it's like a one drug, right? And so there are other molecular targets where the one drug, at least the one that's available, isn't enough, by itself, but maybe it could be enough if you combined it with something else, or even combined with a more kind of traditional therapy. Immunotherapy is like a really still new field. So there's a lot there that hasn't been discovered yet or investigated yet, but combination immunotherapies, are interesting as well. And this is one of the phrases that was said a few times that I thought was very compelling. That what got us here won't get us there, right? So not just more of the same thing. We need new stuff. So these were my key takeaways. That we're kind of maxed out on old chemotherapies, so we need all new stuff. And there's a lot of cool opportunities right now. New ways to do profiling, liquid biopsies, a lot of excitement, immunotherapy, and other kind of new agents. And a lot of emphasis on collaboration, especially when we're thinking about those advocacy parts. I think we're good collaborators in general, and that we can really lean into that. So there was a nice closing. Charlie Roberts gave a nice kind of closing speech, and just really hopeful and forward looking. The science is really accelerating, and we're just all pushing through this like difficult moment that we're in together, where we don't know about funding or how anything's going to work. But there's a lot of good energy there, and we're hopeful that hopefully some of this will come together for osteosarcoma patients and families in a more meaningful way. Yeah, that was a great meeting. And if anyone's interested, I think we're thinking the next meeting will be in Canada next year, but we don't have the location yet. And that they will then alternate between North America and the US, or sorry, North America and Europe going forward.
Okay, great. And probably the same kind of timing each year, late September, or
I think so. I mean, I don't know. Yes. I mean, as far probably.
Yeah. Thank you. That was a fantastic wrap up. So any other, I guess, like across the sessions, and I mean, some of these are, obviously this was all pediatric cancers, not just sarcomas or bone cancers, but kind of just like like epigenetics, it sounds like is kind of a area. Any other kind of emerging new areas that there's a lot of excitement around?
Yeah, I think epigenetics, immunotherapy, and liquid biopsy. Everybody's excited about that. Yeah. Antibody drug conjugates is another area that's sort of new that people are getting excited about too. Not like as much kind of talk about it as some of the others, but yeah, there's a lot of movement right now.
Yeah. And for the immune stuff, I mean, there's, the CAR T, there's NK cells. Was there any particular, I don't know, area that seemed kind of there was the most activity in, and also maybe kind of more translatable for osteo?
Not that I know of. I should say, like, this is not really my area, so I'm not the right person to ask. But it's working well. The delivery is complicated, right? So like, there's finding the right target, and there's a lot of people working on that, and then there's also a lot of other people that are working on, how we give it to the patients. It's pretty intense. How do we monitor and care for the patients, that are receiving immunotherapy? With the CAR T cell therapy, is a lot. um, yeah, and so there's a lot of people working hard on that, and I'm not the right one to answer that
I know that
question.
I was just as that came out of my mouth, I realized that was like an unfair question for you.
It's okay. There's plenty that I don't know, and I don't mind admitting.
um, I was gonna. Oh, the other thing actually about okay, so there's like the epigenetics, but I feel like another area that there might be a lot of focus on. I'm wondering like how much conversation was around this was in like addressing resistance and
Mm-hmm.
like modulating the tumor microenvironment and kind of
Mm-hmm.
that, and probably having to do that in combination with something else, but
Mm-hmm. Mm-hmm.
still that being
Yeah,
kind of it.
there wasn't a lot about that in this meeting, and not because those areas aren't important. It just was like, the focus was really around kind of new different stuff. There was a little bit of mention of kind of single cell sequencing as like an approach, which I know is relevant to the tumor microenvironment work.
And so actually one last question for you about and focusing on the molecular
Yeah.
because of all these different kind of multi-omic strategies. But one challenge that is kind of I don't know is it unique to bone cancers is the samples, like the small biopsy samples, and there have
Mm-hmm.
been challenges in the past about how the samples are processed. So kind of how to kind of address that in this new world of multi-omics and trying to learn as much as we
Yeah,
can
I
from.
think it is really challenging that what's unique to osteosarcoma and like some of the other bone tumors like Ewing is the decalcification and sort of the nature of the location of the tumor sort of within bone, right? Just that the bony matrix itself is challenging. But a lot of the almost all of the pediatric cancer groups are facing the challenge of small biopsies. The brain tumor, community talks about this a lot because they take out little pieces. Of course, it's like the brain, and then they do a lot of molecular profiling, so they want to send it to 10 different places. Especially they're doing methylation profiling pretty routinely. So it is very challenging, and I think that's one of the things that we think about in the sort of molecular lab space is always trying to make our tests better so that we don't need to ask as much of the proceduralists and of the tissue samples that we get, because there are a lot of needs. And we can't forget the microscopy too, right? Somebody needs to look at it under the microscope and see what it is and make the diagnosis. So we want to make sure there's enough for everybody to do all the work that we need to.
Yes. Well, thank you so much for all your work on this conference, which sounded like there was a lot of interesting discussion, and always for your work on kind of making it possible for pediatric patients to have access to molecular testing. And thank you for joining us today to discuss all of that and for always making it better for pediatric cancer patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast player. Marketplace on the MIB Agents channel. A new episode of our Osteo AYA podcast is going to drop on October 16th, next Thursday, or actually in a couple weeks. It's become an annual tradition for our co-hosts Mia and Sammy to have some of our junior advisory board members on as guests, and they share spooky sarcoma stories, which are often cringy, but also usually pretty hilarious. So tune in for a peek behind the curtain into the patient experience and for some laughs. And then we'll be back with OsteoBites on October 23rd. We're going to be joined by Dr. Kelly Makielski and Dr. Jaime Moriano from the College of Veterinary Medicine and the Masonic Cancer Center at the University of Minnesota. And they're going to discuss their work on comparative extracellular vesicle biomarkers for osteosarcoma risk and prognosis. You can find our OsteoBites lineup for the next few months on our website. Please subscribe to our channel so you never miss an episode. And if you have any ideas for future topics you'd like to hear about, please email us at events@mibagents.org. Thank you again so much, Dr. Church, for spending time with us today. And thanks to all of you for taking time out of your day to join us on OsteoBites. We hope to see you back here in a couple of weeks when we chat with Dr. Makielski and Dr. Modiano about the extracellular vesicles. Thank you, Dr. Church. Thanks, everyone.
Thank you.